Hello friends, this is the class on the fermentation. So if you have watched my previous videos, I have already gave my lecture on the glycolysis and the gluconeogenesis and the tricarboxylic acid cycle, and then I gave my lecture on the production of the acetyl CoA from the pyruvate. Now if the oxygen is not present, if the oxygen is not present, then the pyruvate undergoes fermentation, and that means the final electron acceptor is organic substrate, and these organic substrates can be alcohol or lactic acid. This is another way of using the pyruvate apart from using it to make acetyl CoA and then to introduce that acetyl CoA into TCA cycle. Where there is no oxygen present or lack of oxygen, there we find this kind of mechanism where pyruvate undergoes a fermentation in absence of oxygen. And another thing of the significance of the fermentation is that all those NAD plus that has been used in the glycolysis or in the TCA cycle to produce NADH plus H plus, that went to the electron transport chain to produce the ATP, they will they will regenerate, means the NAD plus will regenerate from the NADH plus H plus by the process of this fermentation, so that this NAD plus will be available for again to use the NADH during the glycolysis. Otherwise this glycolysis pathway will be stopped if the NAD plus factors are not available. So now I will start with the lactic acid fermentation that occurs in human. This is the glucose molecule. This is the 6 carbon molecule, and after the glycolysis 2 molecule of 3 carbon pyruvate is produced. This is a this pathway is glycolysis. This pyruvate can produce the acetyl CoA by the help of the pyruvate dehydrogenase complex, and then this acetyl CoA will go to TCA cycle when there is oxygen. That means this glycolysis and then this acetyl CoA production and this TCA cycle, all these things happens when the oxygen is present. That means in aerobic respiration the final electron in aerobic respiration this things takes place. Now when in the anaerobic condition, means the fermentation means in the fermentation not this anaerobic condition is not the fermentation, because in anaerobic condition the final electron acceptor is nitrate or sulfate, not oxygen, whereas in aerobic condition oxygen is the final electron acceptor, and in case of the fermentation the organic substrate like alcohol, lactic acid are the final electron acceptor. Now here in this case when I am dealing with the fermentation, then this pyruvate undergoes a reaction by the help of the enzyme lactate dehydrogenase. The enzyme is lactate dehydrogenase, and this enzyme helps in giving the hydrogen from the NADH to this pyruvate to produce lactate, and now after giving the hydrogen the NADH get oxidized and it again produce NAD plus, which can contribute into the glycolysis pathway again. Now this where this hydrogen has been introduced, look at this carbonyl group in the pyruvate. After the reduction it produces alcohol. If we have followed this organic reactions, the ketone group or the aldehyde group, when they are they get oxidized, they produces carboxyl group. When they get reduced, they produces this hydroxyl group. So this is the structure of the lactate, and this is the lactic acid. Now I should go to my next slide, and there I want to discuss this Cody cycle. This is an important cycle because this lactate which is produced during the fermentation, when the oxygen is not present, this condition arises in the skeletal muscle. When we do the exercise, then the more that during the exercise, the amount of oxygen that we need, sometimes we don't get that much of oxygen there. This fermentation takes place, and this lactate is produced. Now there is no enzyme that is present in the muscle cells which can reproduce the glucose or the carbohydrates from the lactate. So the lactate comes into the blood vessels, and then straight away reaches the liver, where there is enzyme present, lactate dehydrogenase in the liver, which again do the kind of a back reaction, where lactate produces pyruvate. In previous reaction we had seen that pyruvate produces lactate. Here in this in liver, this same lactate dehydrogenase produces pyruvate, and from that this glucose 6 phosphate, this is the opposite path of the glycolysis. It is more or less like this gluconeogenesis. This glucose 6 phosphate is produced, and then the glucose is produced. It is the pathway of the gluconeogenesis. Now after this glucose 6 phosphate is produced, this glucose is produced by the glucose 6 phosphatase, or it can produce glycogen. I will discuss the synthesis glycogenesis in my next video, but before I really discuss that thing, you should know that this glucose 6 phosphate can be used to produce this glycogen. This glycogen can be stored in liver as well as in muscle cells. So now this glucose 6 phosphate again generates this glucose, which comes into blood vessels, and then they again reaches the muscle cells, and they again start this reaction towards the lactate synthesis. And this whole process, this whole cycle is. Now the next fermentation is the alcohol fermentation. Alcohol fermentation. This kind of fermentation can be seen in certain bacteria which takes pyruvate as their pyruvate, and produces ethanol as a byproduct. They metabolize pyruvate, and when we consume ethanol, then we do just the opposite pathway of this, means we go to this direction to remove ethanol from our body. So now this pyruvate, which is the end product of the glycolysis, it is a 3 carbon structure, and the enzyme pyruvate decarboxylase enzyme produces acetaldehyde. Now from the name of the enzyme you can understand that decarboxylation means one carbon will be detached from this structure pyruvate here, and this carboxyl group has been removed in the form of the carbon dioxide. So, and one proton, this H plus ion, has been added to this empty hand of the carbon and forms this acetaldehyde structure. Now this acetaldehyde undergoes a reduction reaction where this alcohol dehydrogenase enzyme plays an important role. From the name dehydrogenase, you can understand that one hydrogen has been added to this acetaldehyde to produce ethanol. Now NADH plus H plus comes into play as a source of this hydrogen, and it gives the hydrogen to the acetaldehyde, and this ethanol is produced. And at the same time, after the hydrogen has been donated from the NADH plus H plus, NAD plus has been produced. Okay, now it will again go back to the glycolysis cycle, and where this hydrogen has been added, look at this structure, there is a double bond in between this carbon and oxygen. And as we have already discussed that when we reduce the carbonyl group, we get the hydroxyl group, and here is the hydroxyl group, and this is the ethanol. So if we want to take an overview of this pyruvate, that is, after the glycolysis, this glucose produces two molecule of pyruvate. Now this pyruvate produces acetaldehyde by pyruvate decarboxylase, and this acetaldehyde again produces ethanol by acetaldehyde by alcohol dehydrogenase. And here one NADH plus H plus NADH plus H plus has been introduced, which will introduce the hydrogen into this acetaldehyde to produce the ethanol, and this NAD plus again will join this glycolysis pathway. Now this acetaldehyde again can produce acetate by acetaldehyde dehydrogenase, and this acetate can produce acetyl CoA by acetyl CoA synthetase, and this acetyl CoA can be used in the lipid production. So we are not much interested into this pathway, but what I want to say here this about this reaction, that sometimes the pyruvate produces, or the pyruvate produces oxaloacetate, means this pyruvate or phosphoenol pyruvate, which are the key product of the glycolysis, they undergoes a kind of a reaction to produce some reactant involved in the tricarboxylic acid cycle, and this kind of a reaction is called the anapleurotic reaction. I should go to the next slide and we'll write it down. This is called anapleurotic reaction, or you can also call it the replenishing reaction, replenishing reaction, or you can also call it the filling up reaction. Basically this kind of reaction takes place to give the reactant component supplying to the tricarboxylic acid cycle. I can give you some examples, like this pyruvate, the structure is CH3 C double bond O minus. This is a 3 carbon structure. This pyruvate, in presence of the CO2 and ATP, produces oxaloacetate. This is CO minus C double bond O CH2 COO minus, and the enzymes involved here is the pyruvate carboxylase. The carboxylase means addition of the carbon into a structure, and decarboxylase, this kind of enzyme are responsible for taking out the carbon from one structure. Another important reaction that I want to write down is this that this phosphoenol pyruvate, the structure is CH2C. Here is one O. This is the phosphoenol pyruvate, and this one, in presence of bicarbonate and the help of the enzyme phosphoenol pyruvate carboxylase, it produces oxaloacetate again. The structure is same here. This is the structure. Now both of this oxaloacetate that has been produced from the pyruvate or from the phosphoenol pyruvate can undergo this tricarboxylic acid cycle to get combined with the acetyl CoA to produce citrate. Now another example of this anapleurotic reaction is this pyruvate. This is the pyruvate structure. This pyruvate can undergo a reaction in presence of this bicarbonate and the NADPH, in presence of the enzyme malic enzyme. It should be malic dehydrogenase, and produces malate. Malate, the structure is like this. The structure is like this. This is malic enzyme, is malate dehydrogenase, and as because this NADPH is there, so it will give the hydrogen and it will leave as a NAD plus and will join the glycolysis to again become reduced and to produce NADH plus H plus. So now again this carbon has been reduced to produce this hydroxyl group. This is the carbonyl structure and this is the hydroxyl group, and I have told you several times that this carbonyl and this aldehyde group, when they get reduced, they produces this hydroxyl group. So what we have done till now is this, that we have already discussed about the alcohol fermentation, and we have talked about the lactate fermentation, and then we have talked about the Cori cycle, and here in this last slide we have talked about the anapleurotic reactions, or you can also call it a replenishing reaction or the filling up reaction, where we produce the reactants of the TCA cycle in a different way, so that we can contribute this into this cycle and get this. If you have loved my videos, if my videos helped you, so you can visit my website, that is easyscience.weebly.com, for more videos. You can find this video in YouTube, and this is the link, and you can also subscribe my channel, and you can also write to me. This is my email address, mukHop89@gmail.com. You can send me your feedback or suggestions or any kind of questions you have. Thank you for watching my videos, and stay tuned for